Today we will study the steps involved in the formation of notochord, mesoderm and coelom in the frog embryo. So before that we will just go to frog gastrula, a late gastrula in cross section. We can see this is the ectoderm which is differentiated into the neurectoderm and the epidermal ectoderm. And here you can see the prospective notochord which will form the notochord of the embryo. This is the mesoderm on either side of the notochordal tissue, and this structure, it is called as a chorda mesodermal mantle. So this is a narrow sheet which is stretching along the longitudinal axis of the embryo, and this is formed by the involution of notochordal cells and mesoderm during the gastrulation process, because it was already mentioned that during gastrulation there will be first the invagination of the endoderm followed by involution of the notochord as well as the notochordal tissue as well as the mesodermal tissue. So because of the involution of the notochordal tissue and mesoderm and the following events in gastrulation, this notochordal tissue plus mesoderm will form a sheet which is seen towards the longitudinal axis of the embryo on the dorsal side, and this is called as the chorda mesodermal mantle. So by the time during gastrulation there is a stage where the blastopore closes. So by that time what happens is that the chorda mesodermal mantle, it will separate from the endoderm. So this is the endoderm, and this chorda mesodermal mantle is now separate from the endoderm. Though the involution started with the endoderm, now the chorda mesodermal mantle is separate from the endoderm. The mesoderm is slowly taking its position between the endoderm and the ectoderm. Here you can see the black shaded region is the mesodermal tissue which is occupying its position between the endoderm and the ectoderm. So this region, it is the prospective notochord. So first, during the when the neural tube formation occurs, simultaneously another event also happens, that is this chorda mesodermal mantle. This chorda mesodermal mantle, it will delaminate or it will split into the precaudal plate in the middle and the mesoderm on the sides. So the first step, the change that is associated with this mantle is that it will delaminate or it will get layered into a precaudal plate in the middle and the somatic mesoderm on the side. So this is the precaudal plate, and this is the this one. This is, these two are the somatic mesoderm on the sides. Now we will move on to the process. So here, this is a cross section of the embryo in this stage. That is, here you can see the neurulation process is initiating. That is, this region which is the neurectoderm, as we saw from the earlier diagram. So this is the neurectoderm, will thicken to form the neural plate or the medullary plate, and this will form a tube. This will form a groove first, which is called as the neural groove, by the formation of the neural folds, and they will form the neural tube. All these can be seen in the video explaining neurulation. So simultaneously at that time, here you can see the precaudal plate, the same precaudal plate which we saw here. This is the precaudal plate. So here what is happening to the precaudal plate? The cells of the precaudal plate, they will expand quickly and arrange themselves into a cylindrical rod-like structure. So it is now free from the mesoderm. This is mesoderm on either side. So it is actually delaminating. Earlier you can see it as a continuous sheet, but here you can see that the notochordal tissue is separate and the mesoderm on either side is separate. So a delamination has occurred, and the cells of this precaudal plate, they expand and arrange themselves into the cylindrical rod which is called as the notochord. Though I mentioned cylindrical rod, this is a transverse section. That is why it is having a circular outline. This is actually a rod-like structure which will appear circular in outline in the transverse section. And this notochord, later fluid-filled vacuoles will appear in this notochordal cells. There will be appearance of fluid-filled vacuoles or vacuolation occurs. Thereby this notochord becomes a turgid structure in the median axis of the embryo. So now this becomes a turgid rod-like structure, and they get surrounded by a sheath. It gets surrounded by an elastic and collagenous sheath. A fibrous sheath will surround the notochordal tissue. So in a later stage you can see a well differentiated notochordal tissue which is surrounded by a sheath, and it is separated from the mesoderm. So here you can see it is separated from the mesoderm. So this is a later stage. Initially they were seen closer. Delamination has occurred. The notochord attains its own structure. It develops a sheath. Now you can see it as a distinct notochord which is covered by the collagenous sheath. And since this sheath is very fibrous and tough, now the cells, they cannot expand much. They can only slide and interdigitate in the long axis of the sheath. They cannot expand in the outward direction. So the cells inside this notochordal sheath will now only slide and interdigitate. So what happens? There will be lengthening of this notochord, lengthening of the notochord. And you can see a diagram of this frog tadpole, where you can see this region, this one. This one is the nerve cord, the spinal cord, the brain with its part, the developing brain. This is the developing brain, and this is the spinal cord which is seen on the dorsal most part of the tadpole. Just beneath this nerve cord you can see this notochord. So this is the notochord. So the notochord is a rod-like structure. So this is the notochord. This notochord is a rod-like structure which is seen below the nerve cord on the dorsal side of the embryo. So here in this diagram, this structure will form the future nerve cord, and just below it is the notochord. So these are two different views. This is a transverse section, whereas this is a lateral diagram of this tadpole. So hope you understood the positions. So this is the dorsal nerve cord. Ventral to it or below it you can see the notochord, but in transverse section it will appear like this. So in transverse section the notochord will appear like this. This is the developing nerve cord which you can see in the neurulation video. Now when the notochord it detaches from the mesoderm. So now you can see the notochord has now detached from the mesoderm. This was the initial mesodermal tissue. Now it is free from the notochord. What happens is that the endodermal cells, you can see the endodermal cells, on either side, they will grow over and form a new archenderic roof just below the notochord. So for that we will just go to the previous diagram, that is the late gastrula. In the late gastrula, this is the notochord. Now it is forming the roof of this archendron. Now the roof of the archendron in the late gastrula stage is formed by this notochord. But once this notochord notochordal tissue separates itself, this endoderm will grow over from either side, and it will now form the new roof of the archendron. So when the notochord separates from this mesoderm and forms a distinct notochordal rod, this endoderm will grow over from the sides and forms the new roof for the archendron. So here you can see the endoderm on either side has grown over. From this side also the endoderm has grown over, and it has formed a roof for this archendron or midgut just below the notochord. So this is how notochord formation takes place. Now we will go to the formation of mesoderm and coelom. So it was already mentioned that the presumptive mesorder, this is the presumptive mesorder. We will go to the diagram back. So this is the presumptive mesorder, the future mesoderm, shaded in black you can see on either side of the notochord. During notochord formation, it will separate or delaminate from the notochord on either side. So now it is located on either side of the notochord. What happens? They will now grow laterally. That means they will grow to the sides and also downwards. So you can see the mesodermal tissue growing downwards. The mesodermal tissue is growing downwards, and they are growing laterally as well as downwards, so as to occupy the position between the endoderm and the ectoderm. This is the ectoderm, this is the endoderm. It will grow downwards and occupy the position between the endoderm and the ectoderm. So this mesoderm occupies the position between the endoderm and the ectoderm. You can see the mesoderm growing down. Here also you can see the mesoderm growing down, and it will gradually occupy the position between this endoderm and this ectoderm. So until they meet here. So growth occurs from this side as well as from this side, and the growth occurs in between the endoderm and ectoderm, and since growth occurs on either side, at the ventral region, at the mid-ventral region, they will meet. Just below this gut they will meet. Now what happens to this mesoderm? This mesoderm, it will get subdivided into segments in the body. So the mesoderm, you can see the mesoderm is dividing into segments. So just below, the mesoderm divides into segments. We will go to another diagram. So you just imagine the body of this organism to be a train which is having many compartments. So the entire mesodermal tissue will split into compartments or split into many segments. Now we will go to each segment. So again, this is a cross section where you can see this is the newly developed nerve cord, this is the newly developed notochord, and this was the mesodermal tissue. This was the mesodermal tissue. So as I already mentioned, this mesodermal tissue which spreads along the whole length of the embryo will be compartmentalized, or it will be divided into many segments. Now we will come to each segment, one of the segments. Each segment has three parts. The dorsal part is called as the somite. So this mesodermal tissue, this is nerve cord, this is notochord, this is endoderm, this is the gut, this is the ectoderm, and what remains is the mesoderm. So this is mesodermal tissue, this is mesodermal tissue, as well as this is mesodermal tissue. Now these three have their names. So all these are mesodermal tissues. 1, 2, 3. So all these are mesodermal tissues. So we will first go to the dorsal most mesodermal tissue. It is called as somite or epimere. Then there is a ventral mesodermal tissue, this one, which is called as the hypomere. So it is the ventrolateral plate, you can see. This is the ventral and lateral plate. This is the ventral and lateral plate, which is called as a hypomere. And there is another mesodermal tissue here, which is intermediate between the dorsal and the ventral mesoderm, and that is the intermediate mesodermal tissue. So you have a dorsal mesodermal tissue 1, intermediate mesodermal tissue 2, a ventrolateral mesodermal tissue 3. This dorsal mesodermal tissue is also called as somite or epimere. The intermediate mesodermal tissue is called as mesomere or nephrotome, and the ventrolateral mesodermal tissue, it is called as the hypomere or the ventrolateral plate mesoderm. So once again, during the notochord, after notochord formation, the mesoderm will fully separate from the notochord, and in the whole length of the body of this embryo, the mesoderm will be compartmentalized, and each compartment is having three segments, that is the dorsal epimere, the middle mesomere or the nephrotome, and the ventrolateral hypomere. Now we will go further. So we will first go to this epimere or the somite. So this is a more advanced embryo than the previous one. So we are first going to epimere. So this is the epimere. This epimere has now split into, again the cells have now rearranged again into three series of segments. That is, so the epimere, it is actually lying on either side of the notochord. So we will go to the previous diagram. When this is the notochord, this is the epimere. So the epimere is lying on either side of the notochord. this epimere itself or the somite itself splits into 3 segments. So, we will go to that. The 3 segments are, one is dermatome, second is myotome, third one is sclerotome. So, the initial epimere now splits into dermatome, myotome and sclerotome. So, first we go to the dermatome. Dermatome is the outermost narrow strip of somite just beneath the epidermis. So, this is the epidermis. Just beneath the epidermis you can see this dermatome, which is the outermost narrow strip of somite. It is this dermatome which will develop into the dermis of the skin. So, dermatome develops into the dermis of the skin. So, dermis is mesodermally derived, whereas epidermis is derived from the ectoderm. This dermatome gives rise to the dermis of the skin. So, we can say dermis is originating from the mesoderm. Now, the second part of this epimere, that is the myotome. Myotome represents the major portion of this somite. The major portion of the somite is represented by this myotome. This myotome develops into the skeletal muscles of the back. The striated muscles of the back of this embryo is formed from the myotome. Now, the third one, the sclerotome. The sclerotome is that part of the epimere or somite which is very close to the notochord. So, you can see this is the sclerotome, this is the notochord. So, the part of the epimere which is close to the notochord is called as the sclerotome, and this sclerotome will form the skeletal tissue around the notochord. So, this is the notochord. When the embryo grows into an adult, skeletal elements will grow over this notochord. And what is the skeletal element developing into? It will develop into the vertebral column. So, the vertebral column, the tissues of the vertebral column are actually formed from this sclerotome. The sclerotome is lying close to the newly formed notochord. So, the cells from the sclerotome will give rise to the vertebral column tissues, or they will give rise to the skeletal elements which will surround the notochord. So, once again, the mesoderm is differentiated into the epimere or the somite, the mesomere or the nephrotome, and the hypomere or the ventrolateral mesoderm. The somite itself is subdivided into 3, that is the dermatome, the myotome and the sclerotome. Dermatome gives rise to the dermis, myotome gives rise to the skeletal muscles of the back, and the sclerotome, it will give rise to the skeletal elements which will form around this notochord, and that is nothing but the future vertebral column. Now, we will go to the second part of this mesoderm, that is second part of the mesodermal segment, that is this nephrotome. So, so far we discussed only about this epimere which is subdivided into 3. Now, we have to go to the nephrotome. So, this is the nephrotome, that is the second mesodermal segment. The nephrotome, it lies on either side of the somite. So, this is the epimere. So, you can see the nephrotome lying on either side of the somite. This is the somite of this side. Here you can see the nephrotome. This is the somite of this side. Here you can see the nephrotome or the mesomere. And it is this nephrotome which will give rise to the excretory organ or the future kidney of the embryo. Now, the last one is hypomere. So, this is the hypomere or the ventral and lateral plate mesoderm. Now, we will see what happens to this hypomere. So, for that we have to go to the diagram of an advanced embryo. So, here this is the nephrotome from which the excretory organs of the kidney are forming. The nephrotome which we discussed just before, this is the nephrotome. Now, this is this region, it is the hypomere. This is the hypomere. So, in the early embryo, this is the hypomere, but in a later embryo, you can see the hypomere has split into 2 layers, the outer layer and the inner layer. So, the hypomere in an advanced stage will get split into 2. This outer layer is called as the somatic layer or the parietal layer, and the inner layer is called as the splanchnic layer or the visceral layer. So, the hypomere splits into two layers, the outer somatic layer or the parietal layer and the inner splanchnic layer or the visceral layer. You can just see the this somatic layer, it is lying just below this epidermis. So, this is the epidermis formed from the ectoderm. The somatic layer is lying close to the epidermis. So, these two together, that is the epidermis plus the somatic layer, it is called as somatoplure. So, epidermis plus this somatic layer, this epidermis plus this somatic layer is called as somatoplure. Now, we come to the inner layer, that is a splanchnic layer. The splanchnic layer is lying close to the endoderm. When the somatic layer is lying close to the ectoderm, the splanchnic layer, it is lying close to the endoderm. So, the endoderm plus the splanchnic layer, it is called as the splanoplure. So, ectoderm plus somatic layer is somatoplure. Endoderm plus splanchnic layer is called as splanoplure. And now we come to the formation of coelom. So, what is meant by coelom? Coelom is a body cavity or the cavity between the body wall and the gut wall. That is what is called as a coelom. So, if you consider an organism which is having a tube within a tube body plan. What do you mean by tube within a tube body plan? You take the case of frog itself. So, we will first take the case of a pen. A pen is having an outer tube as well as an inner tube. The inner tube forms the refill of the pen. So, you can see it is a tube within a tube. The inner tube is the refill which contains the ink and the outer tube which forms the body of the pen. Just like that, in the body of these higher organisms, it is built up on a tube within a tube body plan. That means the inner tube is the gut. So, this is the inner tube or the gut, and this is the outer tube. This is the outer tube. So, this is the inner tube and this is the outer tube. Now, if we again go back to the example of a pen. So, the pen is having an outer body and an inner refill tube which is containing ink. There is a space between these two, the outer body and the inner ink containing space. There is a space between the inner tube and the outer tube. So, similarly, in the body of this organism also, there will be a space between the gut wall and the body wall, and that place is called as the coelom. So, coelom is the body cavity. It will occupy the space between the gut wall and the body wall, and it is actually formed from the mesoderm. So, how is it formed? So, you can see the hypomere, it has split into two layers, that is the somatic layer and the splanchnic layer, and you can see a space between the two. You can see the space. This space. It is this space that will develop into the coelom. So, the coelom is developing from the space between the somatic and the splanchnic layers of this hypomere. You can see, though there is a space, at their free ends they are united. At the free ends these two layers are united. This is a somatic layer which will unite with the splanchnic layer at the free ends. But in the center you can see this cavity. It is this cavity which will develop into the coelom. Now, these two will fuse together. These two will fuse together. In the mid-ventral line, the lateral mesodermal plates will fuse together. You can see the lateral mesodermal plates are fusing together in the mid-ventral line, and these lateral cavities, they will also fuse. So, here there is a fusion, and the lateral cavities also undergo fusion to form a single continuous coelom. Later, in a later stage, these two cavities also unite to form a continuous single coelom which is called as a splanocoel, and this cavity will later expand to form the body cavity of the adult animal. Even this splanocoel, it is this cavity which also gives rise to the pericardial cavity around the heart. So, you can see it is this mesodermal tissue. The mesodermal tissue called as the hypomere is giving rise to the coelom, and coelom is formed from the space between the outer and inner layers of the hypomere. The outer layer is called as the somatic layer, inner layer is called as the splanchnic layer, and the space between these two will develop into the body cavity by fusion. And this cavity occupies the space between the gut wall as well as the body wall, and it is lined by mesoderm as well as covered by mesoderm. So, a true coelom is that which lies between the body wall and the gut wall. So, the coelom is lying in between the gut wall and the body wall, and it is lined as well as covered by mesoderm. So, such a coelom is called as a true coelom. So, this is how coelom develops in frog.